The implantation starts within the and on the day 7 and 8, differentiation of the trophoblast into cyto and syncytotrophoblast. Differentiation embryoblast into epi and hypoblast. And it form a bilaminar germ disc and formation of a amniotic cavity. This occurs in the day 7 and 8. Again, one day passed, sun sets and rises again, and starting of the closure of the that enters the penetration defect by fibrin coagulum, appearance of the lacunar spaces in Syncytotrophoblast, appearance of the Hughes membrane, and formation of the primary yolk sac. Day 11 and 12, the fusion of the lacunar spaces into larger spaces and formation of the Syncytotrophoblast trabeculae, erosion of the maternal vessels, so that the uteroplacental circulation now starts, because lacunar spaces are filled with maternal blood. And same time there is a formation of the extraembryonic mesoderm, followed by formation of the extraembryonic coelom, and which divides into somatopleuric and splanchnopluric layer, formation of the connecting stalk, and formation of the chorion. Day 13 and 14, most important, primary villi is formed. What is primary villi? This above is the epiblast, below is the above is the epiblast, and yellow color is the hypoblast, which forms a Hughes membrane to form a primary yolk sac. And above there is an amniotic cavity, which is lined by Cytotrophoblast and this is the Cytotrophoblast and this is the Syncytotrophoblast, and this is the extraembryonic mesoderm. Ultimately here it will form the extraembryonic coelom. Now in the Syncytotrophoblast, spaces appear, larger space, which first there is a small spaces, ultimately these three spaces, these spaces join to form a larger spaces, these spaces join to form a larger spaces. These we call the lacunae. And the maternal, it is the endometrium, the blood, Syncytotrophoblast erodes the endometrial wall and blood come to fill these lacunar spaces. So this is the lacunae, and in between the lacunae there is a strands of Syncytotrophoblast. There is a strands of Syncytotrophoblast. This is the strands or finger projection of the Syncytotrophoblast, right? These we call the trabeculae. Now, just below the Syncytotrophoblast, there is a Cytotrophoblast, which are cellular, cell membrane can be seen. Now this Cytotrophoblast gradually multiply by mitosis and invades. এটা অন্য কালার করি তাহলে সুবিধা হবে. This is the Cytotrophoblast, right? আমি yellow color করলাম. these cytotrophoblast then multiply and invade these trabeculae. Invade these trabeculae. Now the trabecule is now called primary villi. So what is primary villi? Syncytotrophoblast plus central core of cytotrophoblast. Which are as a central core. Central core মানে কি? মাঝখানে থাকে, बीच में. Cytotrophoblast centrally there is an extension of the cytotrophoblast and we now call this is the primary villi. Later, not in 13 to 14 days, this just underneath this, what is this? Extraembryonic, এটা কি? Extraembryonic কি? তুমি বল extraembryonic কি ওটা? হ্যাঁ? This is the cytotrophoblast. This is the cytotrophoblast, yellow color. And beneath it, what is it? এটা কি এই লেয়ারটা? হ্যাঁ? বাড়িতে পড়ছো না। এই লেয়ারটা কি? তুমি একা বলছো, এরা তো বলছে না। ওটা কি এটা? হ্যাঁ? Extraembryonic কি? হ্যাঁ? Extraembryonic villi, fluid কি এটা? হ্যাঁ? Extraembryonic তুমি? Extraembryonic কি এটা? কে বললো? হ্যাঁ? আরে ভয় পাচ্ছ কেন? ভয় extraembryonic তুমি কি বলছো? Extraembryonic কি বললাম? In between the Hughes membrane and the cytotrophoblast. This is a green color is the cytotrophoblast, yellow color is the cytotrophoblast, and this is the Hughes membrane. This is the amniotic cavity. In between these two layers, there is a development of another tissue. We call it extraembryonic mesoderm. So this is the extraembryonic mesoderm. Within this extraembryonic mesoderm, a cavity will appear. A cavity will appear. We call it extraembryonic coelom, which is filled with a fluid. বুঝতে পেরেছো? This is the, within this extraembryonic mesoderm, a cavity will appear, dividing this extraembryonic mesoderm into a splanchnopleuroid and somatopleuroid layer. This is the extraembryonic mesoderm. After few days, this extraembryonic mesoderm will extend into the the core area of the primary villi. If we enlarge the primary villi, if we enlarge magnifying view, আমরা যদি এটাকে বড় করি, কি হয়েছে? আমরা দেখব Syncytotrophoblast বাইরে. This is the Syncytotrophoblast. In between, there is a Cytotrophoblast core. আমি এটাকে বড় করছি, ম্যাগনিফাইং লেন্স দিয়ে দেখলে Microscope, there is an outside, there is an Syncytotrophoblast with multiple nuclei, and this is the Cytotrophoblast, and we call it primary villi. Now, within this Cytotrophoblast, this extraembryonic mesoderm again extend, एर भेतरे ढुके जाए, इसमें घुस जाता है. क्या घुस जाता है? एक्स्ट्राएम्ब्रियोनिक मेसोडर्म. सो एक्स्ट्राएम्ब्रियोनिक मेसोडर्म एर मध्य ढुके गेल. তাহলে সেন্ট্রালে কে এসে গেল? এক্সট্রাএমব্রিয়নিক মেসোডার্ম, সাইটোট্রোফোব্লাস্ট, সিনসাইটোট্রোফোব্লাস্ট. We call it secondary villi. So secondary villi is equal to Syncytotrophoblast, Cytotrophoblast plus extraembryonic mesoderm. তারপরে কি হলো? Next, again after few days, the blood vessel will appear. Here there is a appearance of a blood vessel, artery and veins, appearance of a blood vessel within this mesoderm. A blood vessel will develop within this extraembryonic mesoderm cord. Cord মানে কি? সেন্ট্রালে যেটা থাকে তাকে বলি cord. CORE, cord. কোনো কিছুর সেন্ট্রালি যদি থাকে আমরা এটাকে বলি কোর. যেমন দিস ইজ দ্য ক্যাভিটি, এটা এরকম রয়েছে. সো ডাস্টার ইজ দ্য কোর অফ দিস বক্স. তাহলে এখানে ব্লাড ভেসেল উইল ডেভেলপ অ্যান্ড ইট ইজ নাও টার্ন টার্সিয়ারি ভিলাই প্লাস ব্লাড ভেসেল. So primary villi, Syncytotrophoblast plus Cytotrophoblast. Secondary villi, Syncytotrophoblast, Cytotrophoblast, extraembryonic mesoderm. Tertiary villi, Syncytotrophoblast, Cytotrophoblast, extraembryonic mesoderm plus blood vessels. Blood vessel ওখানেই ডেভেলপ করবে. এগুলি এখন হবে না. এখন জাস্ট 13 to 14 ডেজে অনলি প্রাইমারি ভিলাই আর ডেভেলপড. তারপরেরটা হচ্ছে আসল. বাকি ডেট ইউ নিড নট টু রিমেম্বার. নো ওয়ান উইল আস্ক হোয়েন দ্য প্রাইমারি ভিলাই উইল অ্যাপিয়ার. বাট নাও হোয়াট আই এম টেলিং, দ্য ডে ইজ ভেরি ইম্পর্টেন্ট. দ্য নেক্সট স্লাইড. So 13 to 14 days means second week ends. 0 to 7 first week, 8 to 14 second week. So primary villi develop, second week ends. Next, third week starts. It is the upper view when we view the embryonic disc from the above. So we can see only the epiblast. We cannot see the, so the embryo, the germinal disc or the embryo is now like that. Now this is the embryonic disc, hypoblast, epiblast. It is more or less circular. Now I am viewing this embryo from the epiblast on the upper surface, on the epiblast view from the amniotic cavity. আমরা amniotic cavity দিকে দেখছি. So we are only seeing the epiblast. We cannot see the hypoblast. Can you see? My this hand, you cannot. So this is the view now. Here is the picture. This is the epiblast. Right? This is the epiblast. Now, two things happen here. First, at the one end, I don't know which end. Maybe here, maybe here. It is circular. Maybe here, maybe here. Two layers, epiblast, type of cell, columnar. Hypoblast, type of cells, cubical, cuboidal, bolechi, right? Epiblast cell, columnar cell. Hypoblast cell, slightly smaller, cuboidal. Now, it is the circular, it is the embryonic disc in one to the periphery. Periphery mane ki? Border. Towards the border, any border of the circle, periphery, perimetry. There will be a circular area where the hypoblast cell becomes columnar. Hypoblast cell is normally cuboidal, but in this area, hypoblast cell becomes columnar and tightly anchored to the epiblast cell. এমন ভাবে এটা অ্যাটাচ হয়ে যায়, no one can enter between the epiblast and hypoblast in this area. So tightly arranged. শুধু ছিঁড়ে যাবে, তাও এরা ভেতরে কাউকে ঢুকতে দেবে না। It can be tear, but no one can enter in this area between the epiblast and hypoblast in one area. We call now area Prochordal Plate. কি বলি? Prochordal Plate বা Prochordal area বা Prochordal area বা Prochordal Plate. Where the epiblast and hypoblast are tightly anchored in a circular small area. Just after it appears simultaneously, opposite to this area, another area close to the periphery, again this hypoblast cell become columnar and strongly attached to the epiblast. Now, the first area that is appear, that we call the Prochordal Plate or Prochordal area, it will be the head end. It will be the cephalic end. It will be the head end. It will be the cephalic end. Right? সার্কেলের আমরা কোনটা জানি না, এখন কিন্তু যেদিকে প্রিকর্ডাল এরিয়া থাকবে সেটা হবে হেড এন্ড, আর অপোজিটটা হবে টেল এন্ড। হলো একটা সার্কেলের আমরা কোনটা মাথা কোনটা জানি না, কিন্তু যেই প্রিকর্ডাল প্লেট ফার্স্ট অ্যাপিয়ার, ইট উইল বি দ্য হেড এন্ড and opposite side will be the tail end, obviously. And now this circular germ disc is slightly now elliptical. এরকম হয়ে গেল। So this is the head end, slightly larger, this is the tail end. And this we call the tail end where the hypoderm is hypoblast and epiblast cell attached strong together, we call cloacal membrane or cloacal plate. আর ওপরটাকে বলছি প্রিকর্ডাল বা প্রোকর্ডাল প্লেট, যেখানে হেড হবে। এখন আমরা বুঝে গেলাম, নাও উই আন্ডারস্ট্যান্ড that it will be the head, it will be the lower region. Now the day must remember, it occurs at about 14th to 15th day. Now on 15th day, very important, on 15th day, these epiblast cell close to the tail end, a longitudinal streak is specialized, thickened. Streak মানে কি? Linear, any কাঠির মতো জিনিস, this we call streak. This part is epiblast cell, thickened, specialized, and we call it primitive streak. Primitive streak. When appear? Day 15. 15 day. কোন দিকে appear করেছে? Towards the tail end. Primitive streak. And the cranial end, now we can say head end, cranial end. Head means cranial. Cranial means cranium, head. In scientific terminology, head ke amra cranial end boli, othoba head end. This head end of the primitive cell again become proliferate. Proliferate means what? Mitotic division occurs and more cells are accumulated. We call it proliferation. Proliferation means cell division. So this head end, these cells become proliferated, make a globular. Globular means ki? Circular, thickened area. We call primitive node. We call primitive node. Primitive node. So this is the primitive streak, this is the primitive streak, this is the primitive streak, and this is the primitive node. Where it occurs? And the epiblast region. And the epiblast region, right? Now, at the epiblast region, in the primitive node, slight groove, slight depression occurs. Depression, depression means ki nichu. Primitive node previously erokom chilo. Eta primitive streak, this is the primitive node. This is the primitive streak, this is the primitive node. Now, slight depression occurs. So that, এই জায়গাটা একটু নিচে চলে যায়, এটা হাইপোব্লাস্ট, এপিব্লাস্ট এরম হয়ে গেল, তাকে আমরা বলি প্রিমিটিভ পোর। এইবার একটু শোনো, লেখা কিচ্ছু করবে না। এরপর একটা টার্ম আছে অর্গানাইজার। হোয়াট ইজ অর্গানাইজার? You come here in this medical college. Previously you were in school, all are general, not specialized. You can do anything. You may be engineer, maybe medical profession, you may be chartered accountant, maybe MBA, anything. Maybe science, pure science, physicist. But when you come here, giving a examination, you are now going to be a doctor. So this is an organizer. More say, তার চেয়েও বেশি কথা, এটাকে when your stream divides at about 10 or 11, science, arts, 11. So at 11, before 11, you are all equal. You have to read the science, arts, geography. After 11, few students are science, few become arts, few become commerce. Right? So at the 11 examination of 10, decide the future course of your life. Similarly, primitive streak will decide the future generation, what the say, which will be the ectoderm, which will be the endoderm, which will be the mesoderm. How? Now you look. Before 11, you are epiblast cell. Before class 11, you are epiblast cell. All are epiblast cell. পেছনে hypoblast রয়েছে, ওটা ছেড়ে দাও। Epiblast. Now this epiblast cell, I am writing white, all are attracted. Few cells are attracted, সব নয়. Few cells are attracted to the primitive streak. Few cells are attracted to the femoral streak. The epiblast cells are normally epiblast or epithelial cells tightly attached. Few cells are now attachment become loose. They migrate, they divide and migrate. কোথার দিকে migrate করছে? Attracted by primitive streak. Right? They go towards the primitive streak, and at primitive streak they change their characters. ক্যারেক্টার হয়ে গেল, চেঞ্জ করে গেল। কেউ সায়েন্সে যাবে, কেউ আর্টসে যাবে, কেউ কমার্সে যাবে। তাহলে এটা হচ্ছে বাইল্যামিনার জার্ম ডিস্ক, হ্যাঁ, ব্লুটা হচ্ছে এপিব্লাস্ট, ইয়েলোটা হচ্ছে হাইপোব্লাস্ট। Now the cells are attracted, goes to the primitive streak, and few cells, খুব ভালো করে এটা বোঝো, অর্গানাইজড হলো, চেঞ্জ করলো ক্যারেক্টার, and become insinuate. Insinuate মানে কি? যদি তুমি এসে এর মধ্যে ঢুকে যাও। সাপোজ she come here and sits in, ঢুকে গেল, একসাথে বসেছিল কিন্তু এর মধ্যে বসে গেল, তাকে আমরা বলি then we call insinuate. যদি ও এসে এদের মধ্যে ঢুকে গেল, then we call it insinuate. The few cells become changed its character and become insinuate in between the ectoderm and epiblast and hypoblast. ঢুকে গেল। ফার্স্ট কি করে? যে সেলগুলি ঢুকলো, they will replace the hypoblast cell. যত hypoblast সেলকে হাটিয়ে দিল, hypoblast, all our hypoblast are removed and their place are occupied by the insinuated epiblast cell. এরা এলো, এসে সব কটাকে বার করে দিল, দিয়ে এখানে এরাই বসলো। They come, they replace themselves and they sit there. And now we will call them, যারা নতুন এলো, কি বলবো? Endoderm. So hypoblast cell never produce endoderm. Epiblast cell replaces hypoblast cell and form the endoderm. গেল? তাহলে তলের যে ইয়েলো কালারটা, they become endoderm. Not only that, these are also replaced by the endoderm. These cells are replaced by the cell. We are all endoderm. Actually they come from the epiblast. আর সবচেয়ে ম্যাগনিফিসেন্ট যেটা হলো, few cells occupy and remain in between the epiblast and the hypoblast, and they become loosely arranged, fibers are there, ground substance are there, we call it mesoderm. To differentiate it with the extraembryonic mesoderm, we call intraembryonic mesoderm. They occupy between the hypoblast, except in the central region, they occupy between the শুধু সেন্ট্রাল রিজনটা পারবে না, কেন পারবে না আসছে আমি। They occupy in between the ectoderm, epiblast and endoderm, by epiblast and hypoblast. We call it intraembryonic mesoderm. We call it intraembryonic mesoderm. কোথায় থাকছে? In between epiblast and hypoblast cell. They remain all the, এইবারে ভালো করে শোনো, they remain in all parts in between ectoderm and endoderm. But এরপরে আমরা দেখব From the primitive node, a long process ultimately goes in the midline. We call it notochord. We call it notochordal process. Ekkuni ashbo. So midline region, middle region is occupied by the cloacal membrane, primitive streak, primitive node with primitive pit or primitive pore, notochordal process and buccopharyngeal membrane. So intraembryonic mesoderm cannot go in the middle part. ওখানটায় যেতে পারে না কারণ আগেই বলেছি they are in the buccopharyngeal precaudal plate. Precaudal plate ultimately will form the buccopharyngeal membrane. So it is correct. So epiblast and hypoblast are strongly attached. So mesoderm cannot enter. Notochordal process, a strong rod-like process, mesoderm cannot enter. Primitive streak, no question of entering. And cloacal membrane, they are also strongly attached. So mesoderm is not entered in between the ectoderm and the endoderm, in the middle line. Otherwise, in everywhere, between the ectoderm and endoderm, there is a mesoderm. Even in front, eta bhalo kore shono. In cranial to the precaudal plate, এটা একদম ভালো করে শোনো ক্রেনিয়াল মানে কি টুয়ার্ডস দ্য হেড এন্ড মানে আমরা যদি লুজলি বলি অ্যান্টেরিয়ার কিন্তু এখানে অ্যান্টেরিয়ার আমরা বলি না দেয়ার ইজ নো কোশ্চেন অফ অ্যান্টেরিয়ার পস্টেরিয়ার উই ডোন্ট সে অ্যান্টেরিয়ার পস্টেরিয়ার উই উইল সে ইন দ্য ফিটাস ক্রেনিয়াল এন্ড কর্ডাল এন্ড cranial to this blastophore the mesoderm can go and right sided mesoderm is continuous with the left sided mesoderm এখন আমরা রাইট লেফট বলতে পারি যেহেতু প্রিমিটিভ স্ট্রিক বা ক্রেনিয়াল এন্ড this is the midline, so this is the right, this is the left. So, and these region ultimately produce septum transversum will develop here, pericardial area, pericardial bud, heart will develop here. Cranial to the oropharyngeal membrane, right? Hello? তাহলে এবার বলতে পারো এই buccopharyngeal membrane is ultimately or precaudal plate, ultimately floor of the precaudal plate, we call it buccopharyngeal membrane, and it is usually rupture, ফেটে যায় and form the mouth, right, from the mouth. And I have now just told you that from this area, pericardial area, the heart will develop. So, in your body, heart is present cranial to the mouth of your হওয়া উচিত ছিল এইখানে যদি হার্ট ডেভেলপ করে if the heart is developed here this is the mouth heart will be above the mouth তাই কি না কি করে হয় না ভেরি ইন্টারেস্টিং হার্টটা চলে আসবে এখানে বাই ফোল্ডিং আসবো আমি আচ্ছা তাহলে এটা শিখলাম and the term repeatedly come into the examination. So the bilaminar germ disc with the development of the intraembryonic mesoderm become trilaminar, 3 layers, right? তাই তো এরপরে কি হলো remaining epiblast cell, few epiblast cell form the endoderm. Few epiblast cell migrate to organized by the primitive streak become the mesoderm and the remaining epiblast cell ultimately form the ectoderm. So all the 3 layers in our body ectoderm, mesoderm, endoderm are all produced by the epiblast cell. And now the germinal disc is now 3 layers, ectoderm, endoderm and in between mesoderm. And this process of conversion of bilaminar germ disc to 3-layer germ disc we call gastrulation. Gastrulation. তাহলে একটু রিপিট করি। Appears on 15th day, primitive streak, thickened linear band of epiblast in the midline at the caudal end of the dorsal part of the embryonic disc. Dorsal part এখন কাকে বলছি? Epiblast যেদিকে রয়েছে, where the epiblast are lying, and hypoblast যেদিকে রয়েছে ventral part. So primitive streak is a very important buccopharyngeal membrane decides cephalocaudal axis. মানে কি? Cephalo means head end, caudal means tail end. So primitive যখন precaudal plate develop করলো, we can know the axis, which one will be the head, which one will be the tail. So appearance of buccopharyngeal membrane is very important. এগুলি বলে দিয়েছি, একবার দেখে নাও। এই দেখো কি সুন্দর। This is the epiblast cell. Beautiful picture. This is the epiblast. This is the hypoblast. The cells are attracted towards this is the primitive streak. And there is a groove in the primitive streak. Development, আমি অত ডিটেল যাইনি, primitive groove বলা হয়, ছেড়ে দাও। These are attracted towards the primitive streak, changes its character and insinuate between the epiblast and epiblast and form the intraembryonic mesoderm. বুঝতে পারছো পিকচারটা? এপিব্লাস্ট সেলই দুটোর মধ্যে ঢুকে গেল। প্রিমিটিভ নোডের আরেকটা নাম হচ্ছে হেনসেন্স নোড, আর এখানে সেন্ট্রালি দেয়ার ইজ একটা ডিপ্রেসড এরিয়া হয়, তাকে বলে প্রিমিটিভ পিট বা প্রিমিটিভ পোর, যেটা লোয়ার এনিমেলে ব্লাস্টোপোর বলা হয়। এইটা দেখো আরো সুন্দর কালার দিয়ে। Epiblast cell are migrating and form the intraembryonic mesoderm, replacing the hypoblast cell and form another layer, the intraembryonic mesoderm. This is the three-dimensional picture, primitive streak and intraembryonic mesoderm gradually forming in between the epiblast and the hypoblast. তাহলে কি কি করলো? ইনভেজিটেটিভ সেল ডিসপ্লেস হাইপোব্লাস্ট সেল টু ফর্ম ডেফিনেটিভ এন্ডোডার্ম, ফর্ম এ লেয়ার বিটুইন দ্য এন্ডোডার্ম অ্যান্ড এপিব্লাস্ট, অ্যান্ড দ্য লেয়ার ইজ কল্ড দ্য ইন্ট্রাএমব্রিয়নিক মেসোডার্ম, অ্যান্ড রিমেনিং সেলস অফ দ্য এপিব্লাস্ট ফর্ম ডেফিনেটিভ এক্টোডার্ম। বারবার রিপিট করছি যাতে পড়তে না হয়। তাহলে এন্ডোডার্ম থেকে কি ডেভেলপ করবে? আমাদের মিউকাস মেমব্রেন, লাইনিং মেমব্রেন অফ দ্য ডাইজেস্টিভ সিস্টেম, রেসপিরেটরি সিস্টেম, এই হ্যাঁ, an associated structure. Mesoderm করবে everything, skeleton, circulatory system, muscles, excretory system and most of the reproductive system. পরে চলে আসবে, এখন এটা ectoderm, skin, sense organ and nervous system. আমাদের ব্রেন কিন্তু ectodermal in origin. Retina is ectodermal in origin. Gastrulation, where the two-layer germ disc become three-layer. এরপরে আরেকটা ইম্পর্টেন্ট জিনিস, এই মাত্র যেটা বলছিলাম, খুব ভালো করে শোনো। তাহলে এখানে এক্টোডার্ম, এখন আর এপিব্লাস্ট বলবো না, এক্টোডার্ম, এন্ডোডার্ম, মাঝখানে মেসোডার্ম। নাও in between the two layers, means in between the ectoderm and endoderm, from the primitive node, from the কোথার থেকে? প্রিমিটিভ নোড থেকে, হেনসেন্স নোড থেকে, সার্কুলার যেটা সামনে প্রিমিটিভ স্টিকের এরিয়া আছে, এইখান থেকে a process, cell proliferate and a process goes towards the buccopharyngeal membrane. আমরা কি এরকম দেখতে পাবো? না, এটাকে এপিব্লাস্ট লেয়ার থেকে বা অ্যামনিওটিক সেক থেকে উই ক্যানট সি দিস প্রসেস বিকজ it is in between the ectoderm and endoderm. So it is covered by the epiblast cell. আমরা দেখতে পাচ্ছি না। যখনই আমরা এরকম ভাবে যখন স্কিমেটিক ডায়াগ্রাম ড্র করি তখন আমরা কিভাবে ড্র করি এটাকে? এইভাবে interrupted line means it is covered by something. Which one is covering it? Epiblast cell. Why it is covering it? Because these process lie between the ectoderm and endoderm at the parallel to the mesoderm, at the same plane with the mesoderm. Mesoderm placed laterally and this process is lying midline. And this is solid process, solid rod of cells grows on. আমি এটাকে সলিডই আঁকছি এখন বোঝানোর জন্য। এখানে ectoderm we cut and remove displace the ectoderm so that we can see it, and we call it primitive notochordal process. Primitive notochordal process, very important feature of the phylum Chordata. And what is the extent? From the primitive node up to the primitive plate, a Prochordal plate. এই যে রেড কালারটা gradually it extends, এইদিকে তাকাও, gradually in between, this is the ectoderm, this is the endoderm, and it is goes towards the primitive plate, and it is a solid rod of cells we call primitive notochordal process. Now, a central part, it is a solid cord rod, so there is a full of cells. Central part, মানে core cells of this notochordal process gradually destroyed, disappears to form a canal within this process. মানে প্রথমে ছিল এরকম notochordal process, gradually central cell disappear, a canal is formed. A canal is formed. This is the notochordal process. Initially it is a solid. After that central cell disappear and canal is formed. Above it is the epiblast and below is the hypoblast. And this canal we call, which is featured by the yellow, Neuroenteric Canal. কি বলে? Neuroenteric Canal. It continuous with the primitive pit, and primitive pit is continuous with the amniotic cavity, amniotic fluid. তাই তো? ওপরেই তো amniotic cavity রয়েছে, amniotic fluid রয়েছে, and this is the Neuroenteric Canal. Now again another change takes place. The floor of this, floor মানে কি তল, which is directing towards the endoderm. Floor of this neuroenteric canal, floor cells of the neuroenteric canal, cells of the neuroenteric canal which is situated at the floor of the neuroenteric canal along with the endodermal cell disappear. ওটাও ভ্যানিশ হয়ে গেল। So that amniotic fluid is now connected with the yolk sac fluid. এর নিচেই তো amniotic fluid রয়েছে। এই যে ফ্লোর এইখানে ধরো ডেভেলপ করেছে, এটা হচ্ছে প্রথমে এরকম ছিল, সলিড ছিল, এইদিকে তাকাও, সলিড primitive notochordal process, centrally canal is appear, neuroenteric canal, next floor along with the hypoblast disappear. এবং এটা যদি প্রিমিটিভ নোডের কাছে আমরা সেকশনটা নেই, if we take section at the level of the primitive node, এটা হচ্ছে এরকম। এটা হচ্ছে depressed area primitive pore. So amniotic cavity is continuous with the yolk sac cavity, but it is a temporary. After that, again the defect is closed, again it becomes solid. Again it becomes solid rod and we call definitive notochord. And it is actually from the midline axial structure, the vertebral process, vertebra of our body develops. But ultimately at birth, all the notochordal will disappear. Thake na kichui. Shudu in between the vertebra, nucleus pulposus will একটা substance থাকে intervertebral disc, it will remain. It will learn later on. But it is very important to produce the central axis and vertebral body formation. So this is the notochordal process. এইখানটায় দেখো, initially it is a solid cord, then neuroenteric canal, floor is disappear, again it becomes solid and it forms the definitive notochord. But at birth, ultimately, এত কাণ্ড হলো, but all notochordal substance will disappear. But disappear মানে কি নয়, এটা vertebral body central axis তৈরি করতে help করছে। সুতরাং কোনো কিছু disappear মানে ও ফালতু নয়। তাহলে এটা গেল। এই যে primitive note কানেকশন করেছে। Midline embryonic structure that develops in the third week of development from cells of the primitive node or epiblast. এই রিসেন্ট ভিউ ছেড়ে দাও। এখন বলছে axial mesoderm থেকে হচ্ছে। From primitive node to prochordal plate extend. Primitive node in the central there is a primitive pit. From the primitive pit notochordal process or head process solid. Ultimately, notochordal canal or neuroenteric canal, fusion of cells of the notochordal canal with the underlying endoderm, flattening of the cells of the canal to form the, ছেড়ে দাও যেটা বলেছি, টাইম চলে যাচ্ছে। এরপরে একটা লাস্ট আজকের অ্যালান্টয়েস। We are going back to the again the primary yolk sac, then the secondary yolk sac, pinstop. Primary yolk sac becomes smaller and we call the definitive yolk sac or secondary yolk sac. Secondary yolk sac is a very important. A part of the definitive yolk sac, this yolk sac is taken up within the embryo. Actually, embryo is produced from this layer only, right? Embryonic disc. Amniotic cavity will not produce any part of the embryo. But the yolk sac will. We all know the esophagus, stomach has got layers. In the cavity, outside the cavity, there is an mucous membrane which is lined by a epithelial cell, right? For example, stomach is a simple columnar, usually. Then there is an lamina propria, vascularis mucosa, submucous tissue, then there is a muscles, then the serosauroadventitia. But the basis is the lining epithelium and surrounding structure only covers it. The most important part of the stomach is subserved by the epithelial cell, that is the columnar, secretion, etc. So it is the basis. So stomach, what is the origin of the stomach? We all say, sir, it is endodermal origin. Boli. Ki boli? It is endodermal origin, partly correct. Because only the epithelial lining is formed by the endoderm. But other structures, muscles, submucosa are all formed by the mesoderm. Blood vessels, etc. All are important. But the basis is the lining epithelium. This is formed by the endoderm, epithelium. And this is actually formed by the yolk sac. How? Yolk sac gives nutrition as long as the embryo is a bilaminar, trilaminar, etc. After that, heart is produced, lungs is produced, kidney is going to produce. So, it needs immense nutrition which cannot be produced, subserved by the yolk sac only, by in our body. So, it will give by the placenta. And when we when we taken birth, placenta is not there. So who will give nutrition? Nutrition will come from outside. Proximate principles, fat, carbohydrate, protein, vitamins, minerals, water, but all will be taken up by the digestive system, which are absorbed in the blood, goes to the factory in the liver, they produce many substances which helps to gives our energy, our nutrition and formation of enzymes, protein, everything. So digestive system is important for absorption of our foods, absorption of our nutrients. And as yolk sac gives nutrition, still it is remain in our body. A part of the yolk sac taken up within the embryo and to form the lining epithelium of the digestive system. A part of the yolk sac, not all, a part of the yolk sac is taken up within the embryo. हमारे एम्ब्रॉयर मध्य ढुके जाए, हमारे बॉडीर मध्य ढुके जाए, बॉडी के अंदर घुस जाता है to form the digestive system, and from the digestive system a part also forms the respiratory system also, trachea, lungs, bronchi, etc. And the remaining part remains small, ultimately disappear. But there is an but. A part of the yolk sac will go inside the connecting stalk, future umbilical cord. A small part of the yolk sac goes inside, this prolongation goes inside the connecting stalk, mesodermal area, and we call it allantois. We call it allantois. The main part, a part of the allantois during development is taken up to use in the development of our urinary bladder. And remaining part of the allantois usually remain as a ligament we call median umbilical ligament. But if it is a persist, karo ligament holo na, there might be producing of a fluid-full cavity, allantois cyst, which may produce carcinoma. So you must think about that. So it is the allantois. So allantois is a part of the yolk sac, and this part of the yolk sac is used for the development of the urinary system, urinary bladder. এই দেখো yolk sac, a part of the yolk sac used to produce the digestive system and part remain as a allantois. Allantois initially continuous with the in our body, normally urinary bladder, but after that this connection get closed and it becomes ligament. We call the uricus.